You're listening to Medscape's In Discussion series on endometrial cancer, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Ursula Matulonis, Chief of the Division of Gynecologic Oncology at the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School. She is the first recipient of the Brock Wilson Family Chair at the Dana-Farber Cancer Institute. She co-leads the Gynecologic Cancer Program within the Dana-Farber Harvard Cancer Center, the Ovarian Cancer Specialized Program in Research Excellence, SPORE grant from the National Cancer Institute within the Dana-Farber Harvard Cancer Center, and an NCI P01 Program Project grant that is focused on targeting replication stress in high-grade endometrial cancers. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Ursula Matulonis. Welcome to Season 2 of the Medscape In Discussion Endometrial Cancer podcast series. Today, we'll discuss targeting DNA damage and repair and replication stress in endometrial cancer. First, let me introduce my guest, Dr. Joyce Liu. Dr. Liu serves as the Associate Clinical Research Officer for the Dana-Farber Cancer Institute in Boston, Massachusetts, as well as the Associate Chief and the Clinical Research Director for the Division of Gynecologic Oncology at Dana-Farber. Dr. Liu is a medical oncologist specializing in gynecologic cancers, and she is my wonderful and tremendous colleague. Joyce, welcome to the Medscape In Discussion Endometrial Cancer podcast.
Thank you so much for having me.
This is a complicated topic, but I know you're going to take us through it expertly because this is a new type of therapy for endometrial cancer, kind of focusing on replication stress and DNA damage and repair. So can you briefly explain the concept of replication stress and its role in DNA repair?
It's a really complicated topic. In really broad terms, replication stress is when the process of DNA replication, which is the process by which DNA is duplicated during the cell cycle, is slowed down and halted due to issues that arise with the DNA synthesis process. So as a concept, replication stress is actually a little bit different from some of the other things that we often think about as molecular alterations or sort of targetable processes in cancer. For example, when we talk about homologous recombination deficiency, we're really talking to the inability of a cell to perform a specific process, which is homologous recombination, that ability to repair double-strand DNA breaks. In the case of replication stress, though, it's actually more of like a secondary downstream outcome. So the replication fork is slowing or stalling, and it turns out this is a result that can be caused by a number of different processes. Some of the most common causes of replication stress are, in fact, unrepaired DNA damage. So as you can imagine, if a cell's trying to replicate its DNA and the replication fork's going along and then it encounters an area where there's unrepaired DNA damage, the replication fork can't progress through that area of unrepaired DNA until the DNA is actually fixed. So to do this, the replication fork has to be stopped. It has to stall or slow down until that area of DNA can be repaired, and then it can progress through that area. So that is how DNA or defects in DNA repair can lead to increased replication stress. So that's one of the ways that replication stress can happen, but there's other causes of replication stress. For example, there's something called oncogene-induced replication stress, which can happen when there's amplification or overexpression of oncogenes such as RAS or MYC or CCNE1. And some of these oncogenes can contribute to DNA repair defects. So that's one of the ways they can cause replication stress. But additionally, oncogenes can actually drive a replication stress by causing things like more frequent or aberrant origin firing. And when I say origin firing, this means they're sort of opening up those areas of the DNA to sort of open those pockets for replication. And so more of the DNA is just trying to replicate itself at the same time. And this in turn leads to things that are called collisions between the replication machinery and the transcription machinery. This then causes the DNA to have to stall again so that replication can go on. And when there's a lot of origin firings, you actually deplete the nucleotide pools that you need for DNA synthesis. And so when you don't have enough materials, then you can't sort of replicate your DNA at the same speed. So that also slows down the DNA replication process. So there's a lot of different causes. I really touched on two of the major ones. There's many more beyond that, but you can see that replication stress is actually something, as we think about it conceptually, that's a downstream effect of a number of different causes, one of which it turns out to be DNA repair.
Yeah, it's a great explanation. It is complicated. And I think every time you hear it more, it gets ingrained in us. So, the next question would be after that great explanation is how important do you think DNA damage repair and replication stress is in uterine cancer biology?
Yeah, so this is a really great question. And I think it's one that we've been working really hard here to understand. Ultimately, I think we're going to find that things like replication stress and DNA damage response is going to be really important in at least a significant subpopulation of uterine cancers. Already, we know that uterine cancer is a really heterogeneous group of diseases. They share a single cell type of origin, but ultimately, there's a group of them, they have very disparate biologies, right? You have your DMMR endometrial cancers, you have your low-grade hormonally driven endometrioid cancers, and then you have your higher grade P53 mutated endometrial cancers. There's sort of three big buckets of endometrial cancer that we think about. I think there's still a lot of work to be done in this space, but right now, a lot of the interest in terms of targeting DNA damage and repair and replication stress has been really focused on those high-grade P53 mutated endometrial cancers. There's some really good reasons for this. So if you have the presence of a P53 mutation, it means there's dysregulation of that G1S cell cycle checkpoint, and that can really set up a situation for increased replication stress because cells that have intrinsic DNA damage, for example, oftentimes they're supposed to pause at that G1S checkpoint so that they don't enter S phase in DNA synthesis and replication with unrepaired DNA damage. But if you have a P53 mutation and you've dysregulated the G1S cell cycle checkpoint, the cell is more likely to progress into S phase with unrepaired DNA damage and to have higher replication stress. I think one of the reasons we're also interested in this is because we've really been guided in part by our experiences with ovarian cancer. High-grade serous ovarian cancers have all those P53 mutations as being central to their biology. And so we think that targeting DNA damage response with drugs like PARP inhibitors have been so effective in certain ovarian cancers that we think that endometrial cancer might be a place where we could see some activity as well. But I think it's really important to remember that uterine cancers are molecularly really different from ovarian cancers. About half of the high-grade serous ovarian cancers have homologous recombination deficiency, but the fraction of endometrial cancers that are HR deficient is actually probably much lower. Uterine cancers, in contrast, are actually oncogenically driven diseases. So, I mentioned earlier about oncogene-driven replication stress. That may actually play a bigger role in endometrial cancer than it does in ovarian cancer.
That's a great entry into talking about more therapies focused on this area. I'm going to credit you, Joyce, with starting the study of targeting replication stress with your original trial of a WEE1 inhibitor a number of years ago. And you can tell us about that and also how you became interested in studying this class of drugs in these higher grade, harder to treat uterine cancers like serous, clear cell, and carcinosarcoma.
Thanks so much, Ursula. I mean, our experience with the WEE1 inhibitors has been really one of the exciting areas that I've seen develop sort of career-wise. I can't take sole credit for it. The idea of targeting WEE1 in uterine cancers actually came from a conversation that our colleague, Panos Konstantinopoulos, and I were having together about what were sort of new therapies we thought we could develop in the endometrial cancer space. We've been talking about replication stress and how does WEE1 really play into it? WEE1 is a kinase that has this key role in regulating the G2M checkpoint of the cell cycle. And if we go back all the way to high school biology, there's actually several cell cycle checkpoints that help the cell regulate its progression through the cell cycle. Two of the most important ones are the G1S checkpoint and then the G2M checkpoints. G1S is also known as the restriction checkpoint. It's basically where the cell does sort of like a systems check before it goes into S phase and triggers the whole idea of cell division and becoming two cells from one. And P53, as I mentioned earlier, has a major role in regulating this checkpoint. At the same time, the G2M checkpoint, where WEE1 is one of the key regulators, is where the cell does its final check of how well replicated is the DNA before it enters M phase, where it actually enters mitosis and starts the process of physical division. So what the WEE1 inhibitors are really doing is they're releasing control of the G2M checkpoint. We got really interested in this in endometrial cancer because we were hypothesizing, as I mentioned, this was a conversation I was having with Panos. And we actually hypothesized that cells that didn't have very good G1S checkpoint regulation and which were intrinsically under high replication stress would become really dependent upon the G2M checkpoint. And the reason is because they would go through their G1S checkpoint sort of unregulated. They would end up in this process of trying to replicate their DNA, and they would be sort of under a lot of stress to do that in time. And so they would be very dependent upon that G2M checkpoint to stop them from entering mitosis before all of their DNA replication was complete. Cells that enter mitosis with underreplicated DNA or unrepaired DNA damage often will then undergo mitotic catastrophe because they're trying to line up the DNA in the metaphase plates. And as the DNA is trying to divide, it can't divide properly during the cell division process. And so basically the DNA kind of falls apart. And so you have these mitotic catastrophe events. And so we really thought that these cells that might be in high risk for having underreplicated DNA or DNA damage would become really dependent upon the G2M checkpoint. When you look at the molecular features of these high-grade uterine cancers, especially uterine serous cancers, uterine carcinosarcomas, almost all of them have p53 mutations. And then what we saw that was that in addition to that p53 mutation, which sets the stage with that G1S checkpoint dysregulation, a lot of them also had other features that would suggest that they had high underlying replication stress. So they would have RAS alterations or MYC alterations, CCNE1 amplifications, FBXW7 mutations. FBXW7 regulates MYC and CCNE1. So we hypothesized that, therefore, these were cancers that would have a vulnerability to targeting of the G2M checkpoint with a drug like a WEE1 inhibitor.
That's great. And again, I think it's really pretty amazing that you and Panos came up with this idea a number of years ago. And then you started off with a drug called davasertib, performing a phase 2 trial that's now been published. And then now there are a number of other WEE1 inhibitors in clinical trial testing now. I think you've already talked about the importance of this class of drug, but I think it would be great to hear from you about that initial experience with the davasertib, and then what are some other WEE1 inhibitors in a clinical trial right now, and where you think WEE1 inhibition as a therapeutic modality is going.
I think it's been a bit of a tricky time in the development of WEE1 inhibitors in the endometrial cancer space, and I'll mention that in a moment. Just sort of looking back at it historically, when we first launched that trial of davasertib in uterine serous cancer, it was really a proof of concept phase 2 trial that we launched based upon a hypothesis where we had this idea based upon the molecular alterations and the mechanisms of how we knew WEE1 inhibitors and davasertib worked, but we didn't have really evidence that this was going to be active. So in that trial, it was really gratifying to see that hypothesis play out in a clinical setting. We enrolled 34 patients with evaluable disease over the course of about a year and saw a response rate of just a little bit less than 30%, with that response really being quite durable, being about 9 months, and patients with a median progression-free survival of just over 6 months. And to put this in context, we conducted this trial back in 2018, 2019, before lenvatinib and pembrolizumab were available, before the antibody drug conjugates were sort of, moving into this space. And so the standard of care alternative at that time was basically chemotherapy. And chemotherapy, as we all know, is just not very active in this setting. It has a response rate of somewhere maybe 10 to 15%. So this was really exciting to see. We then, based upon that, launched a next trial, which was an international phase 2 trial. And that trial accrued a little bit over 100 patients. And it was interesting because we found the response rate was very similar. It was about 26%, but the duration of response didn't hold up the same way. It was only about 4.5 months or so, and the median progression-free survival was a lot lower. It was just a little bit under 3 months. And there's a lot of questions for me about why that might be. One of the things that we really saw in this trial was that the treatment intensity was much less. And I think that's one of the things that you see when you go from a very controlled single center setting to something that's much broader and international, is that patients may not have been able to stay on the drug the same way because of some of the side effects they experienced, most notably decreases in blood counts as well as fatigue. Ultimately, adavosertib was deprioritized by AstraZeneca for other reasons. They were focusing on other parts of their portfolio. But as you mentioned, there's other companies that are developing drugs targeting replication stress, and WEE1 now. Zentalis is one of the leaders in this area. Their WEE1 inhibitor, azenosertib, that they've been developing in ovarian and endometrial cancers. And we saw a nice presentation in ovarian cancer just last month at SGO with their activity in platinum-resistant ovarian cancer. And there are other sort of companies that are developing WEE1 targeting agents. Some of them are dual agents looking at WEE1 and PKMYT1. I think that ultimately WEE1 is really tantalizing as a target. I think that the biology is really interesting, and we have proof of concept that it can be a really important target for certain cancers. But the therapeutic window is something we're going to have to be very aware of, and I think is going to be a major player in how well we're going to be able to move these drugs, at least in their current form, into the clinical arena.
Very well said. Thank you so much. I really want to talk about kind of a potential biomarker for responsiveness to WEE1 inhibition, and that's cyclin E1 amplification. And certainly in uterine cancer, we will sometimes pick up cyclin E1 amplification in serous carcinosarcomas, sometimes clear cells, certainly in some high-grade serous ovarian cancer as well. But I think there has been some kind of uncertainty moving forward about how to measure that. Should we be measuring it via true amplification, and exactly what copy number would be sufficient to cause something amplification, or through immunohistochemistry measures, which is exactly what the azenosertib trial that was presented at SGO did, where they probably captured a higher proportion of ovarian cancer patients with that IHC marker, rather than focusing on copy number. So any thoughts about where that biomarker field is going?
Yeah, it's really interesting. So just for context, right, CCNE1 is the gene that encodes the protein cyclin E1, and cyclin E1 is the regulatory subunit for the CDK2 cell cycle kinase. And basically, the cyclin E CDK2 complex facilitates that G1S transition. So it moves cells from G1 into entry into S phase. And so cyclin E overexpression has been observed in a number of different cancers and really helps to facilitate premature entry of cells into S phase. So it increases replication stress, which is why it's been so interesting as a potential biomarker for WEE1 inhibitors and other drugs that target replication stress. There's a number of ways that you can have high levels of cyclin E, which is the protein in the cell. So one is amplification of the gene, so CCNE1 amplification. And this is something we see in a portion of endometrial cancers, most commonly those high-grade P53 mutated endometrial cancers. We see it in ovarian cancer as well as some other solid tumors. CCNE1 amplification almost always results in cyclin E overexpression. So it sort of is a direct linkage. At the same time, we also know that you can have high levels of cyclin E in the cell, so high cyclin E expression that's completely independent of CCNE1 amplification. So the protein is at high levels, even though you don't have extra copies of the gene. And that can be detected, as you said, by immunohistochemistry. In endometrial cancer, I think it's not as clear what the clinical significance of this is. So in ovarian cancers, we were just talking about the presentation at SGO last month. They presented CCNE1 or cyclin E IHC as a biomarker for response to azenosertib, and saw a difference in terms of response rates when you had cyclin E high by IHC ovarian cancers versus those that were cyclin E low. But we don't have that same level of data in endometrial cancer. And I will say that when we did the adavosertib trial, we did try and look at things like CCNE1 amplification and cyclin E levels, and it wasn't clear there was a clear correlation at that point. I think there's a lot to learn still, But again, that biology between ovarian cancer and endometrial cancer may not be exactly the same. And so the biomarkers for response may not be the same between both groups.
Yeah, agreed. I think there's a lot of work to do. I just want to briefly get your take on the role of PARP inhibitors in the treatment of endometrial cancer right now. And do you think there is a role for these drugs in endometrial cancer?
So right now, the answer is I don't know where PARP inhibitors are going to end up in the endometrial cancer space. I think right now I'm not routinely using them. I think, again, the biology between ovarian cancers and endometrial cancers is very different. Ovarian cancers have a lot of homologous recombination deficiency. That's not necessarily the same as endometrial cancer. And one of the lessons I really took from ovarian cancer is that when we use PARP inhibitors without a biomarker, we may not be doing the best for all our patients. And so one of the things I've really struggled with the data that is starting to come out with adding PARP inhibitors to immunotherapy and the DUO-E trial results where numerically the outcomes look better with that combination compared to IO alone. That study was not statistically designed to actually compare the arms, is that it's not clear there's a biomarker and I don't understand the biology. And so from my perspective right now, without having a better understanding of the biology, without knowing biomarkers for activity, without knowing that there aren't any worrisome signals for the long-term effects, I don't right now feel like I am able to say that the PARP inhibitors today have a place in endometrial cancer. I think it's possible that down the road, there will be at least a portion of endometrial cancers where PARP inhibitors may have a place.
Yeah, I agree with you. I think it's too early. And, I think, like you said, for ovary cancer, we learned that we can actually do some harm in those HRP tumors. So agreed. Briefly, can you talk about your program project grant, which is focused on replication stress and endometrial cancer? Any results to report?
Well, we're in early days still. We don't have results quite yet. But I think that, as is probably becoming clear in our conversation, and as you know, we've become really interested based upon the results we saw with adavosertib and all the work we've done with whether or not we can leverage the presence of replication stress in endometrial cancer to bring new therapies to our patients. And so we have this program project grant that's really focused on targeting replication stress in endometrial cancer. It's been a really rewarding collaboration with the groups here, including with you, Ursula. And the questions that we're asking, I think, have been focused around a number of different topics. One is, I've talked a lot about replication stress as a concept, but the truth is we actually don't have a good biomarker for it. Nobody's ever been able to say, here's what replication stress looks like, and this is what shows a cell is under high replication stress. And even though we've hypothesized that it's the presence of high replication stress that leads to sensitivity to WEE1 inhibitors, we actually haven't shown that in a direct manner. So one of the things we're trying to do in this program project grant is really draw that connection more clearly, understand if there are biomarkers there that we could leverage to see when we can target replication stress. One of the projects being led by Dr. Konstantinopoulos, so by PANOS, is looking at whether we can combine other replication stress targeting agents like ATR inhibitors with PI3 kinase pathway inhibitors, really sort of leveraging that. And then a third project is seeing whether by targeting replication stress, we can actually create a more immunogenic environment and increase the effect of immune-based therapies within endometrial cancers.
Yeah, no, it's been a great grant and lots more to come as we go into year 2. So lastly, can you tell us about any therapeutic targets or treatments that target replication stress in endometrial cancer that you're really excited about?
Yeah, well, I'm still excited about WEE1. All the caveats we've talked about aside, I think that ATR inhibitors, which have really been sort of an area of a lot of development in the solid tumor space, are drugs that could be very interested in terms of targeting of replication stress, potentially in combination. There are drugs that target a parallel to WEE1, which is PKMYT1. And that is actually really interesting as well. It also regulates the G2M checkpoint. The lead compound in that space, unfortunately, has been recently deprioritized, but there's other ones that are in development. And so I think PKMYT1 is going to be a really interesting target. There's, I mentioned earlier, these dual inhibitors that people are developing of the combination of WEE1 and PKMYT1 together. Those are just entering phase 1. Again, that therapeutic window and how well they'll be sort of tolerated will be a bit of a question. And then I think there's going to be, the degraders that are starting to come in that really are going to allow us to hone in on the particular proteins of interest. So WEE1 degraders, CDK2 degraders. These are, I think, all going to be really interesting in seeing if we can tease out the ability to target replication stress while creating a window of tolerability where we don't run into the same hematologic or other side effects that have to date made it harder to bring those agents forward in clinical development.
Joyce, thank you so much for your thoughts, for your insights, your knowledge. It's always great to chat. Thank you so much. It's great to really focus on this topic, which I think is really not so emerging anymore, but is well on its way. So thank you so much.
Thank you for having me.
Today, we've talked to Dr. Joyce Liu about the role of replication stress and DNA damage and repair in endometrial cancer, talked about different potential therapeutic targets that are in testing right now and also in the future, as well as upcoming treatments that are focused on replication stress and DNA damage and repair in endometrial cancer. Thank you so much for tuning in. Please take a moment to download the Medscape app to listen and subscribe to this podcast series on endometrial cancer. This is Dr. Ursula Matulonis for the Medscape In Discussion Endometrial Cancer podcast.
Thanks for listening to Medscape's In Discussion Endometrial Cancer podcast series with our host, Dr. Ursula Matulonis. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on endometrial cancer.